Good evening, everyone, and welcome to our first episode of the new 2020 season of the UT Southwestern Science Café. We're so glad to be back on air with you all, and for our regulars, welcome back with us. And for all of our new guests, we're so pleased to meet you this evening. I'm Jenny King, and I direct public affairs here at UT Southwestern. And on behalf of my colleagues, Corey Tobian and Mindy Sass, and our guest speakers, Dr. Richard Dewey and Dr. Bhavya Shah, thanks for joining us tonight. Science cafes are online conversations where speakers take you in deep dives into science topics. And we want to have fun while we learn. The format's casual, interactive. Speakers are both UT Southwestern faculty and occasional invited special guests. We encourage you to ask questions and engage with us during the program. This evening, we're discussing a very interesting topic, the science of HIFU, High-Intensity Focused Ultrasound, and targeted treatments of the brain, with both Drs. Dewey and more on them in just a moment. As usual, before we begin, we have a few technical matters to mention, so here we go. We are recording the program as well as live streaming it tonight on our UT Southwestern Twitter page. We ask you to mute your microphones to help with audio for everyone unless you are called on to ask a question. And we do encourage you to utilize the chat feature to list questions for both of our doctors. We will start Q&A at the conclusion of both presentations. Corey will be facilitating Q&A and Mindy tonight is her trainee. More on that later. Finally, just a reminder, while we cannot answer personal medical questions, we would love to hear from you with general questions. And with that, I'm very pleased to introduce our presenters. First, we have Dr. Richard Jr. He's professor in the Department of Neurology, director of the Clinical Center for Movement Disorders here at UT Southwestern. Dr. Dewey earned his medical degree with high honors at the College of Medicine in Houston and completed his medical internship at St. Joseph's Hospital, also in Houston, and his neurology residency at the Mayo Graduate School of Medicine in Rochester, Minnesota. He also completed a movement disorders fellowship at the Mayo Clinic in both Minnesota and Arizona. He was recruited to UT Southwestern in 1994 to start the Movement Disorders Clinic, which has since grown to include full-time faculty members. His primary research interests are in the areas of new therapeutics, biomarker discovery, disease modifications of Parkinson's disease. A frequent lecturer and author, Dr. Dewey is a diplomat of the American Board of Psychiatry and Neurology, a fellow of the American Academy of Neurology, and a fellow of the American Neurological Association. And in 2018, Dr. Dewey was included in the D Magazine Best Doctors list, as well as named a Texas Monthly Super Doctor. And we do think he's super. Also tonight, we have Dr. Bhavya R. Shah. He is an assistant professor of radiology at UT Southwestern and a member of our Neuroradiology division. He holds a secondary appointment as assistant professor of neurology. His primary clinical interest is in HIFU. I can't see. Pardon me. His primary clinical interest is in high-intensity focused ultrasound for movement disorders. Dr. Shah is an inventor, researcher, and avid traveler. He holds a bachelor's degree in biology, biomedical engineering, and philosophy from Washington University in St. Louis. He earned his medical degree through the combined training at the University of Missouri School of Medicine in Columbia and completed his radiology residency at Boston Medical Center. He then obtained advanced training through National Institutes of Health funded T32 fellowship program at Stanford, during which he also completed a biomedical device innovation program. He subsequently completed a fellowship program in neurology at the University of Chicago Medical Center. Dr. Shah holds 5 patents and 3 recently granted patents for an array of medical devices. He is the co-founder and chief medical officer of Chimeric Advanced Medical Devices in Palo Alto, California. Dr. Dewey and Dr. Shah, we welcome you to Science Café. And Dr. Dewey, the virtual program is yours. Podium, I always say program, the virtual podium.
All right, very good. Well, it's a pleasure to be with you tonight. And I believe you have the first poll that you're going to show. So we're going to be talking tonight about a couple of clinical questions. And the first one I want you to think about is essential tremor. And does anybody know someone with essential tremor? So you have a couple of options here. I know someone with essential tremor. I have essential tremor. I don't know anyone with an essential tremor, or I don't know what essential tremor is. So let's have everybody vote and just select one of those items and hit submit, and then we'll see a little bit about the audience we have tonight. All right, and I'm not a bit surprised to see this, that most of you actually said that you know someone with essential tremor. And we'll learn in just a few minutes why that is not surprising. And it really relates to how common this disorder is. The next poll, it talks about the other condition we're going to talk about today, which is Parkinson disease. And so this is a test question. The question is, the early signs of Parkinson's include tremor, rigidity, slowness of movement, difficulty with walking, or all of the above. So let's see how we vote on that one. All right, and 89% of you said all of the above. So I know I'm dealing with a sharp group tonight, and it'll be a pleasure to go through this information with you. So let me start. We're going to talk about both diseases tonight, Parkinson disease and essential tremor. And let's begin with Parkinson disease. You may be interested to know that this is the second most common neurodegenerative disease in the world. Only Alzheimer's disease is more common. We estimate that it affects about 1% of the general population over the age of 60. And it is now known to be due to the progressive loss of brain dopamine content because the neurons that make dopamine degenerate in this disorder. Now, the dopamine neurons which degenerate, we now know from recent evidence, is due to the accumulation of an otherwise normal brain protein called alpha-synuclein. And so what happens is this protein, which all of our brains have, for reasons we don't understand, begins to aggregate into small clumps of alpha-synuclein. And those clumps of alpha-synuclein are toxic to certain organs within the cell. In particular, they damage mitochondria, which are an energy pump for each individual cell. And as the energy declines, the cells are no longer able to survive and cell death occurs. And as the cells that make dopamine die out, eventually the dopamine levels in the brain begin to fall. Now, a little bit about the pathology of Parkinson's disease. What does it look like? Hopefully you can see my mouse moving here. But what I'm showing you on the far left-hand side is a section through the human midbrain. And what you see are these dark bands that are visible to the naked eye. There are no stains involved here. And this is what a cut section through the human midbrain would look like in a normal person. If you focus your microscope on those black lines, what you see are these large cell bodies of neurons that are richly populating that area. By contrast, in the brain of Parkinson's disease, you can see that the black lines are disappearing. And if you focus your microscope there, you see very few of those large cells, and the little tiny dots that you're seeing are the glial cells, which are the supporting structure of the brain. Here on the right-hand side, the animation is showing you that what happens is the cell projections, what we call the axons of the dopamine neurons, begin to die back. And as they die back, the amount of dopamine in the brain begins to fall. And that's what leads ultimately to the symptoms of Parkinson's disease. Now, this slide illustrates the epidemiology of PD, or how common this disorder is. There are two ways that we measure it. One is in prevalence, which just means how many people actually have the disease. And what I want you to notice is that in every country that we've listed here, it's going up from 2005 to our estimate in 2030. And much of that is, interestingly enough, being driven by an increase in prevalence in China. And that's happening because the population in China is aging. And we see that on the right-hand side, which is a graph of the incidence, that is the new onset of patients with Parkinson's, graphed across the age spectrum. And what this shows is that the disease is very rare in patients under about 40 or 50, but starting in that fifth decade of life, between 50 and 59, we start to see Parkinson's becoming common, and then it takes off like a rocket. And as we get older, the frequency of Parkinson's, of new cases, begins to go up dramatically. Now, here are the motor features of Parkinson's. We mentioned this in the poll slide, and you all did a great job with it, but resting tremor is probably the most characteristic. Almost everybody knows what a resting tremor looks like, and if you don't, I have a videotape I'm going to show you, but this occurs in about 70% of patients and can really involve almost any part of the body, the face, the jaw, the legs, and of course the hands. Bradykinesia, which means slowing down of movement, is even more common. That occurs in 80 to 90% of patients and is oftentimes one of the most disabling symptoms. And then rigidity basically means that when the physician moves your arm or moves your leg, we feel an increase in resistance, and that occurs in over 90% of patients. So these three are considered the cardinal features of Parkinson's disease, and these tend to be present early in the clinical course. The fourth one on the slide, postural instability, is describing a balance problem that develops, and fortunately that's a later finding. We don't see that in very early Parkinson's disease, but with the progression of time, postural instability can become a problem and can be a major problem. So here's an example of a patient with mild to moderate Parkinson's disease. I want you to look at his right hand as the video plays.
You wish to know all about my grandfather.
And what that is it's a resting tremor. Notice that every time he moves the hand, the tremor gets better because he's taking it out of rest. And that's the key to making a diagnosis of a Parkinsonian tremor.
Why does he care?
In addition to that, the hand tapping test shows that he's slow in executing this movement. So that's the bradykinesia you're seeing there. And you'll notice it has a predominance on one side of the body, which is very common in Parkinson's patients.
Spread the fingers apart.
And again, you're seeing very little tremor when he's holding his hands out, because the tremor of Parkinson's disease occurs at rest. Now, there are a number of drugs that have been developed over the years to treat Parkinson's disease. I don't intend, you'll be happy to know, to go over this list with you tonight in any detail. I show this to you only to make the point that we need so many different drugs because the management of Parkinson's with drugs, while very successful early in the course of the disease, as the disease progresses, it becomes harder and harder to control the symptoms with drug therapy alone. And that's why the pharmaceutical industry keeps trying to come up with new drugs that will help us. Unfortunately, with all of the drugs that are available, we still run into limitations in our treatment and may have to move to more advanced therapies. As I say on this slide, I estimate that about 15% of patients with Parkinson's have a tremor that's severe enough that we can't get it to respond to any of the drugs or even the combination of the drugs that I showed you. And therefore, what we term drug refractory tremor is a major unmet need. And almost every week, I'll see a patient in clinic who is reaching that point where tremor is a problem and we can't get it under adequate control with medication. The standard approaches to this are twofold. We have a surgical procedure that we've had for a number of years called deep brain stimulation. And then the very exciting new option that we've just now developed at UT Southwestern is high frequency ultrasound. And you'll be hearing much more about that this evening. So let me switch gears now and briefly talk to you about the second entity that you need to know about, and that is essential tremor. This, it turns out, is the most common movement disorder in adults. It presents as a postural tremor. And so unlike Parkinson's disease, where the tremor is present at rest, with this disease, the hands are perfectly at rest without any tremor, but when they use their hands or reach for objects, that's when their tremor is present. This condition is believed to affect 1% of the general population and almost 5% of the population over age 65. So this disease is up to 5 times more common than Parkinson's disease. It is inherited and it does run in families. Most patients will notice that there is a relative that is one of their previous ancestors who had the disorder. And frequently, if we see an older patient, they'll have children that have it. So it tends to run vertically through the inheritance pattern. And what's really interesting to me is that many people never seek care for tremor from a physician. And it's because it runs in their family. And they just figure, well, this is a familial thing and it's normal for me to shake when I'm reaching for objects. And as a result, we physicians tend to see the tip of the iceberg, where patients really have bad tremor and it's become highly disabling to them. Now, for years, most of the time that we've known about essential tremor, we didn't know anything about its pathology. And that's because the early autopsy studies that were done showed no clear abnormalities. And so we would always just tell our patients that this is a biochemical problem in the brain, but not a structural problem. However, there's a famous neurologist who I'm happy to say has just recently joined the faculty at UT Southwestern as our chairman, whose name is Elan Lewis. He has been studying essential tremor for almost his entire medical career. And he's made a number of significant discoveries. And one of those is that in a specific portion of the brain called the cerebellum, there's a reduction in the number of these Purkinje cells, which are the primary inhibitory cells in the cerebellum. And so we now think that it's loss of those cells that is the most likely cause of essential tremor. And this is a slide from one of his recent papers where he did a very clever thing. These little round blobs that you see here that are connected by that green line are the Purkinje cells. And so what he did, he had the idea, if I just measure the distance between those cells, that'll give me an idea of how many cells there are in the brain. And so he did this in essential tremor patients, and they repeated that in normal brains that came to autopsy. And this graph on the right shows you the results. So if you're a control patient and you don't have this type of tremor, you have a whole lot of cells. This is the number of cells on the y-axis, and the distance between them is short, so they're packed in tight. By contrast, if you have essential tremor, you have fewer of the cells and the distance between them is longer. So the bottom line is, this is pretty clear evidence that the number of those cells is dropping out in the cerebellum. And we now know that these Purkinje cells send an inhibitory signal to motor pathways. And so when you lose an inhibitory cell, it disinhibits a neuronal activation that can trigger a tremor problem. This is a slide on the prevalence of essential tremor, and very much like Parkinson's disease, there's an age-related incidence. So it becomes much, much more common as the individuals age. And here's an example of a patient with essential tremor. Notice he has no tremor at all with his hands at rest in his lap. So it's the opposite pattern that you saw in Parkinson's disease.
Not constantly,
But when he reaches his hands out, that's when the tremor becomes a problem. And you can imagine that if you're trying to feed yourself or drink from a glass or apply makeup, that would be a real issue if that hand is going like that.
If you're like this, almost touch your nose, but not quite.
In many of our patients with essential tremor, it's worse when the hand is near the face, which obviously compromises eating, drinking, and doing a number of other activities of that nature. It also can be a huge problem for writing. And oftentimes we have patients referred to us primarily because they cannot write and they need to be able to write for their job. And so this becomes a hugely disabling component of this disorder. And here I've asked him to draw us a spiral on that paper. And you can see that's really a challenge for him to do. Here's a slide of the different drugs that are available for essential tremor. There are many of them. Of the ones on this list, there are 4 major drugs that we will use kind of in sequence in trying to bring this under control. But the truth of the matter is, on the very best case scenario, we can reduce tremor by about 50% with 1 or more of these drug therapies. And if you had tremor as bad as I showed you in the videotape, 50% of that is still bad enough to be disabling. And so we run into issues where we need further help. The current estimate is that about 50% of patients with severe essential tremor get insufficient benefit from drug treatment. And therefore, for them, we're also going to think about these invasive approaches, such as deep brain stimulation, which is the older, more tried and true approach that involves surgery and the implantation of electrodes into the brain, or this new exciting alternative, high frequency ultrasound. And at that, I will stop and turn it over to my colleague, Dr. Shah.
Thanks, Dr. Dewey. Good evening, everyone, and thanks for joining us. Tonight, I'll be talking about high-intensity focused ultrasound, next-generation image-guided therapy. So just a quick disclosure statement that the patients in my talk tonight have signed privacy videos and waivers and been consented for their videos to be shown tonight. So I always like to start my focused ultrasound talks with this slide. I know we're limited in our ability to be interactive, but does anyone recognize what these devices are? When I was consenting my first patient for focused ultrasound, he turned to me and said, "I know you think you're one of the first sites to be doing this, but they've been doing this forever." When he saw the surprised look on my face, he started laughing and said Star Trek. So the first image is a tricorder. It's kind of like a futuristic MRI scanner. It scans the patient and diagnoses them. The second is a transcranial focused ultrasound device treating a clot in a patient with a stroke. As you can imagine, a way to non-invasively treat diseases of the brain has been a fantasy for at least the last 100 years. I'm here to tell you that today, this fantasy is becoming a reality. So how does focused ultrasound work? Well, if as a kid, you ever used magnifying glass to focus light rays onto a piece of paper to light it on fire, you're already a focused ultrasound expert. But instead of using light rays, we're using ultrasound beams. And don't worry, we aren't lighting anyone on fire. Today, high-intensity focused ultrasound is FDA approved for tremor-predominant Parkinson's disease and essential tremor. However, the future is in drug delivery and neuromodulation. I'll showcase some of our work later in the talk. So this is what the equipment looks like. And everyone will recognize the MRI scanner. And on the scanner bed, you have a helmet. When you open the helmet up, you see 1,024 ultrasound probes. Each ultrasound probe sends its own ultrasound beam, which is focused across an intact skull onto a single point in the brain. That point is the focal point, and it's where the maximum amount of energy deposition takes place. You can see is that you move away from the focal point, the degree of energy delivered drops dramatically. We monitor this with real-time MRI thermometry. We obtain an image before heating and an image after heating and use phase shift MRI to determine the thermal dose that has been used. So how does it work? The patient is positioned inside the MRI for anatomic localization and temperature monitoring. The physician uses the MRI scan to create a treatment plan. The focused ultrasound helmet generates ultrasound waves from multiple trajectories that converge onto a single point in the brain across an intact skull. This technology is currently FDA approved for essential tremor and tremor predominant Parkinson's disease. Identifying the target to treat in tremor patients is very difficult on standard high-resolution MR images. So targeting is done by measuring approximate distances from landmarks. As you can imagine, each one of our brains is slightly different, and so this can be inaccurate by a few millimeters or more. To address this limitation, at UT Southwestern, we use advanced imaging methods that identify white matter tracts within the brain. White matter tracts are essentially cables that connect different parts of the brain. So to identify your target, you just have to know where the cable is in the part of the brain that you're interested in treating. Here's an example why this matters. Here are two images from two patients. You can see both the size and the shape of the target, the white outline, are different. Although additional independent studies will be needed to validate this technology, I think that improving targeting is a critical step in moving this technology forward. Now that we've covered the science, let's look at the patient experience. These are a few before and after clips from patients that we have treated at HIFU at UT Southwestern. We start the morning with a neurological exam immediately before the treatment. This includes a comprehensive tremor assessment and their ability to draw a spiral. We then repeat the neurologic examination immediately after treatment and follow-up visits.
And today?
Today it just works slicker and crystal.
So you can see the treatment responses for yourself in both the immediate post-procedure care and a few days after the procedure.
Wow.
Look at that.
Oh, thank y'all, thank y'all, thank y'all. Everyone.
This is the best part of my job and makes all of the hard work and
Thank
effort
you.
worth it.
Wow. Look. See.
Yay. I could actually get stuff in my mouth without spilling it all over me.
So again, this is the best part of my job. You know, it makes everything worth it, right? So clinical treatments are just one aspect of the UT Southwestern Neuro-Focused Ultrasound program. The Focused Ultrasound program at UT Southwestern is a multifaceted collaboration between several centers on campus, UT Dallas, and collaborators at other centers and my lab. The lab works on a number of focused ultrasound technologies that span from preclinical focused ultrasound to human clinical trials to improving the delivery of focused ultrasound for clinical treatments. We have been fortunate to receive several awards to fund our work. So depending on the amount of energy you deposit, you can change the biological effect of focused ultrasound. So high intensity focused ultrasound destroys tissue, as in the clinical treatments. But when we use the lower energy, focused ultrasound can be used to enhance drug delivery or change how individual neurons behave. The blood-brain barrier is a wall between the bloodstream and the brain. It's an obstacle to getting therapies and diagnostic agents into the brain. We can use focused ultrasound in combination with microbubbles to open the blood-brain barrier. We inject the microbubbles into a peripheral vein. Once they reach the brain, we can expose them to focused ultrasound. Once they're exposed to focused ultrasound, they start to vibrate and they temporarily push upon the blood-brain, open the blood-brain barrier. An example of how we can use this clinically is Alzheimer's disease. Alzheimer's disease is characterized by the abnormal deposition of two proteins, amyloid and tau. Preclinical work has shown that just opening the blood-brain barrier can reduce amyloid protein. One of the clinical trials that we're involved in is using advanced imaging. So this first image right here is an amyloid PET scan, and it shows you where all of the abnormal amyloid protein is deposited in this specific patient. We use this scan to then target areas to open up the blood-brain barrier. After this is complete, we wait a month and repeat the amyloid PET scan. And we try to see if there has been a change in the amount of amyloid that was deposited because of the blood-brain barrier opening and if it cleared. Thanks to some recently awarded funding from a local foundation working to cure Alzheimer's disease, the BVB, we will also be able to add a tau PET scan before and after treatment to understand what happens to amyloid and tau in the brain before and after blood-brain barrier opening in humans. Tau protein is more closely linked to disease progression in Alzheimer's disease. My work with Dr. Diamond in the Center for Alzheimer's and Neurodegenerative Diseases specializes in immunotherapy to identify very small amounts of tau in the brain, even before a patient has developed the symptoms of Alzheimer's disease. When our body interacts with a foreign protein, it makes a large antibody. However, certain animals make very tiny antibodies called nanobodies. This is Bernard. Bernard is an alpaca in France. When we inject him with human tau protein, he makes nanobodies to the human tau. We collect the blood from him and purify these nanobodies. Don't worry, the entire process does not hurt him. We then label the nanobodies with a radioactive tag so we can see it with imaging. If we open up the blood-brain barrier in a transgenic mouse, so a mouse that has human tau in its brain, and then we inject the nanobody after opening the blood-brain barrier, we can see that it lights up. If we use a normal mouse, because it does not have human tau in its brain, there is no signal. This is important because currently there is no way to diagnose preclinical Alzheimer's disease, and diagnoses only happen after significant irreversible damage has taken place. Nanobodies can also be therapeutic because they could be used to remove the abnormal tau from the brain and drain it into the blood. This is even cooler because we can expand this approach to treat other protein deposition diseases. Another therapy we're working on is gene therapy. Gene therapy is a way to change how a cell functions by delivering missing or alternative genes to it. It is usually done with an empty virus filled with the gene you want to deliver. But one of the biggest limitations for gene therapy in the brain are delivery methods. You can inject it directly into the brain, but that puts the patient at risk for injury and bleeding. What we have shown is that we can use focused ultrasound to open the blood-brain barrier and then inject the gene therapy into the blood. So if you look at this images, the red arrow is the side where we've opened up the blood-brain barrier, and this is the corresponding histology image. The dark brown spots show how much gene expression has happened after the blood-brain barrier has opened. And on the other side, the white arrow, we haven't opened up the blood-brain barrier. So if you look at this image, the side where the blood-brain barrier is open, there has been much more gene therapy delivered. You can see when we didn't open up the blood-brain barrier, there was some, but not nearly not as much as there was on the side where we opened up a blood-brain barrier. So this capability plugs directly into the gene therapy program at UT Southwestern, allowing for safer, more targeted, and efficient delivery methods for gene therapy. Thank you for your time today. If I was to summarize why I think neurofocused ultrasound is a next generation technology, it would come down to these 5 things. Number 1, it's non-surgical. Number 2, there's no anesthesia, and as I've shown you tonight, there's an immediate functional response. Number 3, we can do targeted drug delivery. Number 4, we can pair it with advanced imaging, either white matter tracks that I showed you, or metabolic imaging, or molecular imaging, like the amyloid stuff that I showed you. And finally, it allows us to modify disease before it causes long-term consequences or damage. With that, thank you for your time tonight. I have quite a few people to thank, especially my mentorship team, Dr. Chopra, Dr. Dower, Dr. Diamond, and Dr. Malajan, and Dr. Rofsky, as well as all of the support from the President's Office, the UT Southwestern Foundation Office, and my department. Thank you.
Thank you, Dr. Shah and Dr. Dewey. That was fantastic. We are going to move on to audience questions. So please, if you have any questions, please go ahead and list them in the chat. And I'm going to go ahead and start calling on you. When I do call on you, please feel free to unmute yourself, turn your camera on, and ask your questions to Dr. Shah and Dr. Dewey. Our first question comes from our dear friend, Dr. Stu Ravnik, who's actually also our very first Science Cafe speaker. So Stu, please ask your question.
I put 1,000 questions in the chat, so
I know,
I don't know
I'll keep coming back to you, don't worry.
I gotta scroll up and see which one I was looking at first, but fantastic presentation. And I'll just start with the last one first. So A, it's incredible that you can open up the blood-brain barrier. How temporary is that? And B, can you explain to the audience why the viruses you're using for gene therapy are not harmful to patients?
Sure. Yeah. So the blood-brain barrier opening is dependent on the parameters of focused ultrasound that you use. So you can open it anywhere between 2 hours to 6 hours, depending on what you want to do next. And it's
Fantastic.
temporary, so it closes right back up. What makes the viruses not dangerous is that we've basically removed the viral DNA or RNA or proteins that would cause an injury in a human and replace them with the genes or DNA or RNA that we want to actually deliver to the patient.
So those are similar to the nanoparticles that, in some way, similar to the nanoparticles that are giving us all the relief from COVID.
Yes.
Fantastic. Oh my gosh.
Thanks, Stu. Our next question, it looks like who asked it actually fell off just a second ago, but I'm just going to go ahead and ask it. Dr. Dewey, do you ever see patients with both Parkinson's disease and essential tremors?
Yes, unfortunately we do. And it appears now from having looked at this extensively that essential tremor, which as I said is many times more common than Parkinson's, is actually a risk factor for the later development of Parkinson's disease. So if you have essential tremor, you have a slightly greater risk of later developing Parkinson's than you would in the general population. Again, we don't know why that is. As far as we know, it's not the same pathophysiology. Again, in Parkinson's disease, it's alpha-synuclein deposition in the substantia nigra. In essential tremor, it's dropout of Purkinje cells in the cerebellum. So we're not certain of the link there, but it does appear in epidemiological work that it's a risk factor.
Great, thank you. Our next question comes from Ann Quill. Ann, do you want to go ahead and unmute yourself and ask your question? Which actually was one I asked Dr. Shah earlier. I'll give Ann a second. Okay, well, I will go ahead and just read Ann's because it was actually a question I had as well, which was how long do these improvements from the HIFU treatment last?
Sure. So, you know, if you look at some of the studies that have been published on high intensity focused ultrasound, they use the landmark based methods for targeting. So what that means is that they, you know, measured an approximate distance over and treated that spot. They saw a tremor response and they stopped. And so those initial studies show that, you know, immediately after treatment, people have like a 90% response to the treatment. And then if you look at them at 5 years, you know, that comes down to anywhere between 60 and 70%. And just to be conservative, I like to quote 50% at 5 years. But we're using these new advanced imaging methods that map individual brains. And so I think that will also have a big effect in how, the permeance of treatment. So if you're able to completely ablate the part of the brain that's causing the tremor or completely disconnect it, we would expect that you would see, you know, longer term tremor improvement. And that's one of the things that, you know, the group at the Mayo Clinic has reported. It's one of the things that they've been doing as well.
Thank you. And I know that was my question earlier, so and it did come up, Dr. Shah. So our next question comes from Peggy. Peggy, if you want to go ahead and unmute yourself and ask your question.
Can you hear me?
We can hear you. Great.
Thanks. Yeah, just wondering, my mom has MRI, some metal in her head from an injury. And so does this keep her from having the ultrasound as an option?
Yeah, well, it depends on what kind of metal it is, if it's affected by the MR scanner or not. But yes, it could very much so affect the ability to have HIFU if you have metal that can't be in an MRI.
Okay, thank you.
Mm-hmm.
Our next question comes from Vicky. Vicky, you want to go ahead and unmute yourself and ask Dr. Shah and Dr. Dewey your question?
Hi, can you hear me?
We can hear you.
Okay. Well, my question is, my husband was diagnosed with Parkinson's, and mainly it's just a tremor in his left hand that's pretty fairly controlled with medication. But is there any, I mean, he can still feed himself and things, but is there any advantage to starting this treatment early on in a diagnosis to prevent it from progressing?
Yeah, that's a good question. And I would say as a general rule, the answer is no to that. Our approach is to treat patients with the medications first. And if we can bring their tremor under adequate control with medications, we're good. Keep in mind, the tremor doesn't necessarily progress. And one of the fascinating things about tremor and Parkinson disease is that with the progression of the disease, in many cases, the tremor actually gets better. And the problem is the patients develop other symptoms such as slowness of movement, gait difficulty, balance impairment that come to dominate the picture. So our approach would be to offer HIFU or deep brain stimulation surgery to those patients for whom our drugs are not working and the tremor remains disabling.
Okay, thank you. I mean, he still does, he is getting slowness of movement, and, you know, the shuffling and the things like that seems to increase just a little bit. So thank you.
Thank you, Vicki. So I'm actually going to read one of our questions that did come into the email box. Dr. Dewey, can you talk a little bit on, or Dr. Shah, the outlook on finding the cause of Parkinson's?
Well, that is a $64,000 question. Believe it or not, we've known about Parkinson's disease in the scientific literature since 1817. So more than 200 years, we've known about Parkinson's disease. There has been a hard target search for a cause of Parkinson's for at least the last 100 years, and we still have not uncovered it. The best we can say is that we believe there is a genetic predisposition. There's something in the genes that sets you up for being at risk of Parkinson's disease. And then very likely there's an environmental insult that creates the trigger, which ultimately leads to the onset of Parkinson's. And when you have diseases like that have a genetic component plus an environmental component, it's enormously complex to sort it out and to try to find one specific thing. So I'm not highly optimistic that we're going to have a simple answer anytime soon.
Great, thank you. One more question that did come into the email box is, what other diseases are being studied for HIFU treatment outside of Parkinson's and essential tremors?
Right. So with focused ultrasound, you have the high intensity applications. So for that, we can think of things like essential tremor or Parkinson's disease, chronic thalamic pain syndrome. So these are central pain syndrome, where a part of the brain and another part of the thalamus is misbehaving and causing pain. So it would be the same idea, but we're just targeting a different part of the thalamus. Similarly, there have been some clinical trials looking at OCD and some psychiatric conditions such as depression. But on the other side, there's the blood-brain barrier opening side. And so that's really being looked at for delivering chemotherapy or genetic therapy or other things to either brain tumors or certain cells that need some new gene therapy. So that.
Great, thank you. Moving back to our chat box, Shilpa, it looks like you have a question for Dr. Dewey and Dr. Shah. You want to go ahead and unmute yourself?
Are there any adverse effects to this?
For high intensity focused ultrasound?
Mm-hmm.
Yeah, so, for the clinical treatments for treating tremor and in the setting of Parkinson's disease or an essential tremor, because of that part of the brain is surrounded by other very eloquent areas that control movement and sensation, targeting is everything, right? So the side effects would be trouble walking, swallowing, speaking, trouble sensing things, feeling things. That's really what's been reported in the literature. But the way we do the neuro HIFU procedure is that we initially find the target and then we deliver low energy ultrasound. So that low energy ultrasound does a couple of things. One, it tells us if the patient's tremor is getting better. And number two, it tells us if the patient is having side effects. So the patient is not having any side effects and the patient is getting a response from the low energy or low intensity focused ultrasound, then we know that we're in a relatively safe spot and we can go ahead and increase the energy with high intensity focused ultrasound and ablate it. Because we're still giving low energy and sometimes it is not in the right spot, right? So some patients do develop these side effects, but the majority of these side effects resolve at 1 year.
Thanks.
Mm-hmm. Great, thank you. Our next question comes from Tajaji. If you want to go ahead and unmute yourself.
Hi, Dr. Bhavya Shah. Thank you so much. My question is about how HIFU is going to benefit like cancers like glioblastoma.
Sure. So, with HIFU, with the ablation, that's something that needs to be studied a little bit more. But I think for glioblastoma, the role of focused ultrasound is really disrupting the blood-brain barrier and delivering either checkpoint inhibitors or other chemotherapeutic agents to that specific target.
And it looks like you had one more question you had just added about treatment. If you want to go ahead and ask that right now.
Yes, yes. So what's the role of HIFU? Like you said that you can deliver low energy and then keep, is there a way to repeat this in multiple settings where you deliver low energy multiple times for treating the same tremor? Yeah.
Oh, well, no, for the tremor, it's usually that once we find the part of the brain that's causing the tremor, we go ahead and ablate it. Now, there have been instances where patients have come back for a second treatment because the first treatment hasn't been as permittive as they'd like. But really, when you talk about multiple applications of low intensity focused ultrasound, just recently, there was a paper out of the group in Toronto where they opened up the blood-brain barrier once a month for 12 months as part of a patient's chemotherapy regimen. So they would come in the first week of every month, have the blood-brain barrier open, the chemotherapy delivered, and they follow the patient. So that gives us some idea of the safety profile, right, of focused ultrasound in humans.
Thank you.
Mm-hmm.
Our next question comes from Charlandra. Charlandra, do you want to go ahead and unmute yourself?
Yes, I don't think you've answered this question yet. I'm not exactly sure, but are there any risks using HIFU for long-term brain health?
Right. So that's something that we don't really know the answer to. It's a relatively new technology, right? And I always like to think of radiation therapy when I answer this question. For a long time, people thought that there were no other effects from radiation therapy. There are some very specific protocols we follow and some principles, some guiding principles that are kind of important to radiology and in terms of intervention. And one of them is as low as reasonably possible, right? So our lab specifically works on determining how much lower can you go in terms of energy and still get the biological effect that you want, whether it's ablation or opening up the blood-brain barrier, changing the way neurons fire.
So our next question comes from Bobby. Bobby, do you want to go ahead and unmute yourself? Bobby Weil. Okay, just a second. Okay, well, I will go ahead and ask Bobby's question, which are, what are the downsides to HIFU? Is it possible it will also cause trouble with walking?
Well, so that's kind of what we were talking about with the adverse effects, right? So as long as you are well within the part of the brain that you're intending to treat, there are no adverse effects in terms of other functions of the body and the brain.
Great. Our next question comes from Terry. Terry, do you want to go ahead and ask your question? So Terry wants to know, when will HIFU be available for UT Southwestern patients with Parkinson's dyskinesia?
So that's a great question. And I think that there was just a recent study published in the New England Journal of Medicine looking at treating another site for other Parkinson's disease symptoms. So it all comes down to picking a target that is shown to be effective and safe. And so my hope is that targeting structures like the pallido-thalamic tract would be a way to get this to make this possible. I just don't have a timeline on it at this point.
Great, thank you. Our next question has been asked if we would read it. We don't have audio, but they want to know, how can the ultrasound electronics survive the high magnetic field of the brain?
Yeah, so it's actually specifically designed to be compatible in a high magnetic field environment.
Great. Our next question comes from Sapura Yadudu. Do you want to go ahead and unmute yourself and ask your question? All right, I wonder if we're having some trouble with people unmuting themselves tonight. So, is there a patient profile that better lends them to HIFU treatment? Alternately, who would not be a good fit for that kind of treatment?
Right, so right now with MR-guided high-intensity focused ultrasound, the FDA has only approved us to do unilateral treatments. So if you're right-handed and your right hand tremor is really bad, but your left hand tremor is okay, unilateral tremor, that would be a great candidate for neuro HIFU, especially if somebody did not want an invasive procedure. However, if someone had tremor in both hands, it was equally bad, right? And they wanted to seek therapy for both at the same time, then deep brain stimulation would be the better alternative because deep brain stimulation is available to be done bilaterally.
Great. So our next question, and we've actually gotten a couple of these, and Dr. Dewey and Dr. Shah, I'm not sure if this is even one you're going to be able to answer. Is this covered by Medicare and most insurance plans?
Yes. So MR-guided high-intensity focused ultrasound for essential tremor and tremor-predominant Parkinson's disease is covered by Medicare. It's just recently been covered by Aetna, and it's already covered by Blue Cross Blue Shield.
Great. And our next question comes from Angie. Angie Gibson, do you want to go ahead and unmute yourself? I think we're having some unmuting issues. Okay, so Angie wants to know if this works on people with advanced stages of Parkinson's.
Yeah, well, it's one of those things where it hasn't really been tested and tried yet, you know? So I can't really speak to that at that moment. Dr. Dewey might have some more insight into.
Yeah, I would say that, I mean, based on the question, I saw what she chatted in here, where there's issues with significant balance impairment. I would say that that's unlikely to benefit from either HIFU or the deep brain stimulation treatment. Unfortunately, the balance impairment of Parkinson's is not a dopaminergic problem. And that's what makes it so challenging, is that we can give dopamine enhancing drugs for the bradykinesia, for the tremor, for the rigidity, but we don't have good drugs for the balance impairment. And so you would think, well, that's what we need surgery for. Well, we're not there yet either. We just haven't cracked the nut of the balance compromise issue in Parkinson's disease. And so I would say that is a topic for future investigation.
Thank you. We did get a question. A lot of people are asking us to read. Their audio is not working. You had mentioned that there are some studies going on about possibly using this for mental health disorders. Are there any clinical studies that are actively looking at that, or is it more in the future?
No, I think there are some studies, but I think they're in Europe. And I don't know of anything off the top of my head that's in the United States. That's not to say there isn't. It's just that I don't know.
Nothing at UT Southwestern.
Yeah, nothing at UT Southwestern right now.
Great. Dr. Dewey, this question is for you. What is the latest in biomarker discovery that we're seeing related to movement disorders?
Well, yeah, it's a good question. And we are fascinated with the whole issue of biomarkers. We feel that discovery of a major and important biomarker for Parkinson's disease could potentially transform our ability to do clinical trials. The problem is we're struggling to find and validate a biomarker. There are multiple labs that have reported a particular blood compound or CSF compound that seems to track Parkinson's disease. The problem is when we try to replicate that in other labs, we run into problems. So the issue with biomarkers is so much of the attention is on biomarker discovery and not enough attention is being given to biomarker validation. And until you can get a biomarker that can be validated, that it can be done by people in labs all over the world, find the same thing and demonstrate the correlation with the disease progression, you don't have an answer that's suitable. So we are still in that stage of needing to come up with even one good, valid biomarker for Parkinson's.
Well, thank you. That's really interesting to hear. I know there's a lot of biomarker research going on and people hear that in the news. So it's good to hear a great explanation of it. So I have one more question for each of you, and I know this is kind of going to be a little bit of a loaded question before I back to Jenny. But what do you guys think is the future of brain health? Which I realize is probably the most loaded question I could ask, but I know a lot of people are interested in it.
Yeah, well, I tell patients who ask me that or a similar question that the brain is truly the last frontier of medicine. Of all the organ systems in the human body, we know less about the brain than we do about the others, and that's because of its sheer complexity. There are billions of neurons and interconnections, that are so far above our ability to understand them today that we're still scratching the surface. And I tell patients regularly that there's a whole lot more about the brain that we don't know than that we do know. And that's a lot because we know a lot about the brain. And the very fact that we can talk to you about Parkinson's disease and essential tremor and show you this pathology and that nucleus and this tract tells you we know a lot, but there's that much more that we still don't know. So I think the key to brain health in general is good health in the rest of your body, because really that's all the rest of your body is for. It is a brain support structure. That's what your heart is for. That's what your lungs is for. That's what your muscles is for. They all serve the brain and try to keep it healthy. So what we tell patients is eat well, don't drink a lot of alcohol, don't smoke, and above all else, exercise. Exercise is the one thing that we truly believe can slow down the rate of Parkinson's disease, and it undoubtedly contributes to brain health.
Thank you. That was great. I'm going to throw that same question to you, Dr. Shah.
Sure. No, I completely 100% agree with Dr. Dewey on this. I think being healthy, living a healthy lifestyle, and exercising are all important parts of it. But, I think that focused ultrasound does offer us stuff that people have not had in the past, right? The ability to study how neurons or collection of neurons or circuits are behaving together in concert to give normal function, and what happens when we have disease. I think gene therapy is, just exploding right now. I think the understanding of how these networks work together and really studying that and then being able to modulate that either invasively or non-invasively, I think is really the future of brain health and treating brain disease.
Well, thank you both. That was wonderful. I am going to now pass back, Dr. Dewey, Dr. Shah, thank you for your wonderful presentations and all the great answers to questions. I'm going to go ahead and pass back to Jenny to go ahead and close us out.
Hey, awesome. Thank you so much. I wrote down that your body is a brain support structure, Dr. Dewey. I really like that.
I liked that too, Jenny.
It says it all, doesn't it? Thanks to both of you. Tonight was really fascinating, and to know that we have this to help patients and transform lives in North Texas is really inspiring. And I do look forward to UT Southwestern and the O'Donnell Brain Institute doing that important research to kind of break through the last frontier of medicine. So it's very exciting to work on, and even if it's from behind the scenes. And of course, that's my cell phone. We're just gonna let that phone ring. No, I'm not. Shucks. So sorry.
Thank goodness for Zoom.
It's the world of virtual.
I know. Thank goodness for Zoom.
It always
so sorry. This has not been my moment tonight. I've just had a lot of messes here, but back on track. Thanks everyone. Thanks to Corey and Mindy for being our tech gurus, and to Corey for moderating our Q&A. It's always important to make sure we get to as many questions as possible. But for our regulars in particular, I want to say a special word of thanks to Corey for her partnership in this program and share the news that she is departing UT Southwestern for a wonderful new opportunity in the private sector. We would well. I know, and thank her for making Science Cafe an engaging and, we think, successful program. Will be back with us on Tuesday, May 4th for our next episode, which will focus on another high technology treatment, Pulsar, which provides personalized treatments for cancer. And we're going to put that registration link in the chat and also follow up with an email. To everyone here tonight, it is so good to be back with you this evening. As always, look for an email soon with a brief survey about our program. We really value your feedback. And we are now going on to COVID. Phase of the pandemic of getting our vaccines, and our message to you from UT South, please get the shot. Go to utswmed.org, click on the yellow bar at the top, and sign up for a vaccine appointment. Same day appointments. Also shared in the chat a couple minutes ago is information about our next community about COVID vaccines. We have 2 experts in infectious diseases talking about COVID. So join us this Tuesday at lunch. follow-up email will include those details. And even if you are vaccinated, we still need you to wear your masks, social distance, and continue vigilance with hand hygiene. We need to work together to improve the health of North Texas and get us all back to a new normal. And for now, we wish you good health, good spirits, and a good rest of your evening. We are adjourned. Thank you.